A single line on a chromatogram can delay a drug approval by months. Not a failed batch, not a safety scare, just a purity result the FDA reviewer doesn't trust. And most teams don't find out until the deficiency later lands. Hello, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams to build HPLC purity methods that hold up under FDA review. Today we are walking through the 5 HPLC purity testing mistakes that quietly delay approval and how to catch them before a reviewer does. If you have found anything valuable in this channel, please like and subscribe. This gesture of appreciation means a lot to us. Let's start with what's actually at stake. When you file with the FDA, reviewers don't just read your purity results and related substance results. They try to break them, re-examining your chromatograms, impurity limits, and validation data for anything that doesn't add up. When something doesn't, you don't get a rejection, you get a deficiency letter, and your review clock stops. 1 week related substance method can add an entire review cycle. Months lost, launch delayed. Almost every one of these delays traces back to a handful of avoidable mistakes. Here are the 5 HPLC method mistakes that most often delay FDA approval. 1. A method that isn't stability indicating, blind to degradation, products that form on the shelf. 2. Trusting a peak that isn't a pure when 2 compounds coalesce under one clean looking peak. 3. A mass balance you can't explain, where acid drops but impurities don't account for the loss. 4. Treating every impurity as equal, ignoring response factors and ICH thresholds. And 5. Weak system suitability that lets a reviewer question every number you report. Let's take them one by one. Mistake 1 is the most expensive. A stability indicating method must separate and detect the degradation products that form as your drug ages under heat, light, acid, base, and oxidation. Too many teams validate against the clean, freshly made sample and move on, but a method never challenged with the forced degradation stays blind to the impurities that grow on the shelf. When the FDA sees flat stability data with no forced degradation evidence, they don't conclude your product is clean, they conclude your method can't see the problem. Mistake number 2 is peak purity. The second mistake is trusting a chromatographic peak that looks pure but isn't. On screen, you see one clean symmetrical peak and report it as single compound, but 2 substances can coalesce, hide beneath the same peak, and a standard UV detector will never tell them apart. This is where peak purity matters. A photodiode array detector or mass spectrometry confirms one peak really is one compound. Skip that check and you underreport a real impurity or misassign your main peak. The gap between the peaks you can see and impurities truly present is where submissions get challenged. Now, mistake number 3, third mistake is a mass balance that you can't explain. Mass balance means the drop in your assay should be accounted for by the degradants you measure. If potency falls 3% but total impurities rise 0.5%, something is missing. A degradant you're not detecting or one the method lost entirely. Reviewers use mass balance as a fast integrity check on the whole method, and a gap undermines everything else. If your team is wrestling with a mass balance gap right now, drop a comment describing it. Those real cases are what I dig into on this channel. The fourth mistake is treating every impurity as equal. In area percent, a peak is just a peak, but chemically, impurities don't all respond the same way to your detector. If impurity absorbs UV half as strongly as your drug, area percent understands its real level by half, enough to push it across an ICH threshold without you realizing. ICH Q3A and Q3B set 3 tiers: reporting, identification, and quantification, each demanding more work. At Resolve Mass, we have seen sponsors get a deficiency because an impurity crossed the identification threshold that response factor corrected data would have flagged months earlier. Get the response factor right and you report the number that's actually true. The fifth mistake ties the others together: weak system suitability and validation gaps. System suitability is the set of checks before every sequence, for example, resolution, tailing, injection repeatability, proving the instrument was fit to generate the data. Loose criteria or none, and a reviewer can question every result. So here is how the teams that pass build it: risk method design, force degradation, a peak purity check, validation down to reporting limit with clocked response factors, and system suitability tight enough to catch a bad run before bad data ships. So what should you walk away with today's discussion in this video? Prove your method is stability indicating before you trust it. Confirm your peaks are actually pure. Explain your mass balance. Correct for response factors against ICH thresholds, and make system suitability strict, not decorative. Get those 5 areas right and your purity HPLC method data stops being the reason for your approval slips. If you are working on an IND, on an ANDA or biologics program, and you want a CDMO or CRO partner in North America who treats, Resolve Mass Laboratories supports small and mid-sized pharma teams from method development through submission. If you have found anything valuable in this channel, please like this video and subscribe our channel. This gesture of appreciation means really a lot to us. Thank you so much for watching this video till the end. See you in the next one. Until then, goodbye for now.